Next guest in this session is Paul Cisek with a talk, The Long Evolutionary History of the Brain's Functional Architecture. Paul did his Bachelor of Science in Computer Science at Rochester Institute of Technology in New York, and his PhD in Cognitive and Neural Systems at Boston University. And since 2003, he works at the University of Montréal as a professor, and he studies decisions, planning, and movement control. And he's also currently working on a book on the evolution of the nervous system that is resynthesizing our conceptual taxonomy according to the history of evolution, as I actually mentioned in the introduction. So thank you very much, Paul, for accepting our invitation, and we can start.
Well, thank you very much. Dziękuję bardzo za zaproszenie. I'm very pleased to be here, and I'm sorry I missed the first few talks, but I just had a previous commitment. I would have liked to see those. Hopefully I'll see the recorded versions. My talk's going to be probably a bit different, because I'm going to talk about mostly about the things that are innate in a sense of inherited from our ancestors, trying to devise what that is. And I'm really going to focus on the question of functional architecture of the brain, very neuroscience oriented. And I would claim that much of the brain's functional architecture is not learned, that it's actually inherited. And one reason to believe that is if we consider how different are our modern human lives than the lives of our ancestors and the lives of animals in the wild, they're dramatically different. And yet, if we look at the functional organization, it's really quite similar in humans, non-human primates, as well as other mammals and vertebrates even. And what I mean by that is, basic visual processing pathways, things like reinforcement circuits, patterns of activations when you have humans versus animals doing similar tasks, suggesting that there is some basic organization that we share among animals, that despite the fact that what we do with it is so different, actually stays more or less consistent. And of course, that motivates us to ask, well, what is that organization? So that's really the question I'm going to address for most of my talk. Now, cognitive neuroscience suggests it's something like this. That's essentially the organization of brain processing is essentially information processing functions, some of which are perceptual, like object recognitions. Other are cognitive functions like working memory or decision making. Others might be action related, like trajectory planning. And then neuroscience in the recent decades has tried to find the neural correlates and the mechanisms responsible for these different functions. Now, as many of you probably know, it hasn't really fit. That the basic parts list of cognitive neuroscience doesn't really fit so well with what we see in the brain. Now, I'm not really going to go through this too much. I think many of you are familiar with this kind of data, but the basic message is that it doesn't really respect the distinctions that we made on the basis of psychology. And for example, things that should be unified appear to be actually rather distributed. So there is no place where you have a sort of a model of the external world, but instead you have divergence of information. So visual processing diverges into dorsal and ventral streams. There are multiple different representations of the space around us in the parietal cortex, etc. Conversely, things that should be distinct are mixed. So for example, you can decode sensory, motor, and cognitive variables from the same regions, often from the very same neurons, as if these functions are actually mixed in the same cells. Things that would appear to be, you would expect to be serial, appear to unfold in parallel. So for example, you don't see perceptual processing followed by decision-making, followed by action processing, but rather these things seem to happen more or less at the same time in parallel across a highly distributed cortical and subcortical circuitry. Now, perhaps that really shouldn't be that surprising when we consider that the concepts of cognitive neuroscience are based on concepts of cognitive psychology, which was really aimed at a very specific type of behavior, mostly aimed at explaining human thinking. But the brain is, of course, much older. and animals have had these structures for much longer. And that motivates us to ask, well, if cognitive neuroscience doesn't give us the pieces that define the brain's architecture, what are the right pieces? And so I would suggest that one approach for trying to figure out those pieces is to consider the brain's evolution, because I think we all agree that in the end, it's a product of an evolutionary process. Now, doing that, I think, forces us to trying to follow the footsteps of evolution, essentially reconstruct the brain in this way, forces us to confront certain observations. There's a widespread myth when people think about evolution that it's nature's way of finding answers to problems posed by the world, by adapting animals. But that's not really a good description of what evolution does, because evolution doesn't actually identify problems at all. It merely produces variations of some functioning ancestor. And then among those variations, it favors those that happen to accomplish something that used to be a problem. So the problem really isn't identified until it's really solved. It's not really identified at all, you might say. And the important thing to consider here is that the variations that actually are introduced, that enter into the game of natural selection, are actually very limited. For a variation to be selected, obviously it must first be possible. And many variations are simply not possible as a variation of a functioning ancestor. And the reason for that is that the genome doesn't actually describe a blueprint or like a connectome for the brain. It describes a recipe that determines the developmental process that produces the brain. And because, like any recipe, it consists of stages, each of which depends on previous stages, it can't just modify arbitrarily. For example, if you make a modification to an early stage, even if that in principle would be useful, it's going to violate the assumptions upon which later stages are based, producing essentially a big mess and a non-viable animal. So evolution can't really arbitrarily make changes to the developmental process. There's only a few things that it really can do, and that is elaborate and extend the process, and it can differentiate and then specialize different pathways in that process, which means that the kinds of architecture you might see in a descendant is highly constrained by the architecture that was present in the ancestor. So the mechanisms that enter into the evolutionary game are highly constrained by the history and the particular contingencies of development. Now, I would say that this is actually good news, because this makes it possible for us to infer what were the steps in evolution and follow along this path. And furthermore, it actually gives us a kind of taxonomy of systems over the course of history. So I think we can use this to construct a different taxonomy of concepts. So the rest of my talk is going to be really this brisk walk through evolutionary history along the lineage that leads to humans. So here's the phylogenetic tree of animals expanded primarily on the lineage that leads to humans. And here's the various points where we diverge from other animals. The thick lines are where there's fossil data. There's some example animals along these different lineages. And I'm going to follow along essentially this particular path through this history. But I'm going to do this very quickly. It's going to be probably very hard to assimilate most of this stuff, especially for those who may not be familiar with the jargon of neuroscience. So mostly I would like you to note that there's a continuous story of progressive specialization. And I'm going to start with the very origins of life, which there's a general consensus that life began with autocatalytic sets. So these are systems of chemical reactions where some enzyme catalyzes the production of another enzyme, which catalyzes another enzyme, and so on, closing a loop. And once you get that, you get both a type of simple replication as well as simple types of metabolism that maintain this chemical structure. Now, once these systems enclose themselves in membranes, you can now distinguish two types of metabolic control. Control entirely within the membrane. So you control the concentration of some, let's say, necessary nutrients through feedback control. And this is physiology. And evolution will discover this because it exploits reliable laws of chemistry, etc. But sometimes you can't you don't have the machinery to produce a particular nutrient that you need. You have to absorb it from the environment. And to do that, you might need to move around the environment to put yourself in a place where the concentration is higher. And this is behavior, essentially extending metabolic control through the environment through interacting with it. This is a very old idea, back to Dewey, but also Piaget and many others. And evolution will discover these kinds of things because it exploits very reliable laws of nutrient statistics, physics, etc. So from that perspective of behavior as a kind of a control system, it's perhaps not surprising that neurons, when they appeared around here, were as specialization of the external ectoderm of our ancestors. Essentially a specialization of initially pluripotent cells with a both sensory and motor role to cells that were sensory, contractile, and integrative, like neurons. And in the earliest animals, this was a diffuse net across the cup-shaped body, but with two specializations that have been identified by Detlev Arendt and others. An apical nervous system rich in chemosensitive receptors, and a blastoporal nervous system responsible for contractile action to move the animal or ingest water, etc. These animals moved around the world on something called levee walks or levee flights, where there's a period of time when they make small movements, exploiting a local environment, and occasionally make a longer range movement to explore to a new sort of greener pastures. So this is actually a little bit similar to what Alison Gopnik was just saying, but really physically, at a physical level, exploiting where you are versus exploring and trying to find a new environment. And Thomas Hills and others have suggested that this governing between these two behaviors was the original role of the neurotransmitter dopamine, that if your intake of nutrients is high, is at a good rate, then dopamine is high and that motivates you to stay locally. But if it drops because you've depleted local nutrients, dopamine drops and you make a long range movement to explore. So this is a major fundamental distinction in behavior of animals that appeared probably around here. Now you can still see this organization. Oh yes, and also I would like to say that the dopaminergic control was sort of a higher level control from the apical nervous system through hormonal secretions to control the types of activity of the blastoporal nervous system, either move locally or make a long range burst of movement. Now this organization can still be seen in darians like jellyfish. In our branch, the body elongated, the blastoporal fused into a slit to make a digestive tube, and the apical and blastoporal nervous systems merged at one end. This is where the head will appear, and this merging is sort of a precursor of a primordial hypothalamus, high-level controller of metabolism. Now, in protostomes, which includes insects, insects and annelids and mollusks, etc., you still see this organization this way. In our branch, the body inverted dorsoventrally, and the neural plate folded into a tube, creating a basic organization of all chordate vertebrate systems. All of our nervous systems are essentially a tube composed of these neuromeres that have expanded, diversified in evolution since then. That's the basic plan of the brain. Now, this can be seen very clearly if we examine the cephalchordate amphioxus, which gives us a good window into early animals. Essentially concluding that it consisted of essentially a hypothalamus, a high-level controller connected to a spinal cord, with dopamine-dependent search being governed sort of from this higher level to this lower-level controller. These creatures also possessed an escape circuit involving a single photosensitive patch of neurons projecting to a structure called a tectum that projected downstream. So if a shadow fell on this patch, the animal will quickly swim away. We get neural crests and tunicates, etc. I'm going to skip over some of that. In early vertebrates, what happened is this single eye patch split into two that migrated to the sides of the head. And the originally bilateral input became primarily contralateral. And the reason for this is that's useful is now if a shadow falls on your right eye, you're going to first cause contraction on the left side of your body, turning away from the threat. And now you have oriented escape behavior. This expanded further with a kind of retinotopic mapping from locations in space to locations in the escape circuit to create a kind of a map of avoidance actions, oriented avoidance actions downstream. Now, This then specialized further for a part of the tectum with contralateral projections. Now there's a contralateral input and a contralateral output. So now you will turn towards the source of stimulation. This creates an approach circuit. So now there's the specialization that you see, which creates these two types of interaction with the world: oriented approach towards certain things and escape from other things. And the selection between what to do, approach versus avoid, was done through basic combinations of cues, which ethologists call key stimuli. So for example, if something is large and expanding rapidly, you probably want to avoid it. Now, this organization of this sort of contra-ipsi escape system, contra-contra approach system is seen still quite literally in lamprey from the work of Sten Grillner. It can also be seen in zebrafish quite clearly, and it's even in mammals. In fact, all vertebrates have these kinds of systems. They've been retained since probably about 500 million years. Another major innovation of early vertebrates was the expansion of part of the hypothalamus, this alar portion over here, into what would ultimately become the telencephalon. I was surprised to learn this, but it turns out that the entire forebrain, including the cerebral cortex and other structures, are really just outgrowths of the hypothalamus, what was originally the hypothalamus. And the telencephalon consisted originally of two segments: an outer segment, the pallium, which receives sensory input, largely olfactory, but also visual, and a subpallium, which projects downstream to selectively activate these approach and avoidance systems. This will become the basal ganglia. This region also receives a new type of dopaminergic input, phasic dopamine, which says not just that things are good, so stay local, things are not so good, so go somewhere else, but a more punctate reinforcement that says what you just did in this particular state was extra good, so do it again if you find yourself in that sensory state. And so, of course, this was a major innovation because it allowed reinforcement learning. So now you could learn what things to approach, what things to avoid, etc. And the specialization, very early on, there's a specialization in these systems where there's a kind of one system that's specialized for this local activity. When you're in a good environment, you want to exploit it. You want to approach certain things. You want to learn what you want to ingest. And this was governed by this more lateral, ventrolateral parts of the pallium, whereas exploration to new locations was governed by this more medial section. This part here will ultimately become the hippocampus. Now, in jawed vertebrates introduced more sophisticated processing in the hindbrain, leading to what we call the cerebellum. I'm going to go through this rather quickly. Essentially, elaboration of sensory processing here to allow predictive filtering, adaptive filtering of sensory input, such that you can now cancel out the effects of your own motion on your visual and sort of pressure senses, and pick out more precisely the motion of things in the world. And this kind of predictive processing really took off and allowed our ancestors to overcome some of the challenges of swimming fast with a large body and become very effective predators. So you get sharks at this stage. Now, the telencephalon continued to expand. So you see additional parcellation of the thalamic projections to different parts of the brain, the septal nuclei as well as the amygdala, and a kind of a parcellation of the control system, and particularly in this ventrolateral exploitation system, with a sort of more ventral section specializing in learning appetitive cues, and a more caudal section near the amygdala for learning aversive cues, still in the control of local exploitation, while the medial pallium specializes in exploring and navigating and etc. Now, tetrapods, upon coming out on land, encountered a very different world, a very different terrestrial world, with much larger visual range and a wider variety of opportunities for action, or what Gibson called affordances. new demands like thirst, etc., and dramatically expanded their behavioral repertoire. And in concert with that, they expanded, in particular, the pallium. Different subdivisions become increasingly apparent in tetrapods. Now, if we take a slice through it, we see how these things developed quite differently in different branches. So amphibians, for example, they have a great need to stay near the water or they dry out and die, and they expanded primarily the medial pallium, useful for navigation, so they could find their way back to water, etc. Sauropsids, which is the branch that leads to reptiles and birds, they dominated the diurnal world, and they expanded their tactile systems, which primarily guide their visual motor control, but also the regions of the forebrain that receive information from the tectum, particularly the ventral pallium here, which expands dramatically especially in birds, to form the dorsal ventricular ridge. And this has particular specializations for auditory and visual information, etc. Now, and I would say that this is still kind of processing what we might hypothesize for very early animals. The key stimuli that will indicate whether a particular object is something you want to approach or avoid, that then later controlled through downstream. So this is what this aspect of visual and auditory and somatosensory processing did. Whereas this other section here, this other projection of visual information to what is called the dorsal pallium, did something else. And I would propose that this region here specialized in detecting locomotor affordances, so the opportunities that exist in the world, again, with this expanding repertoire. And this doesn't require full-fledged object recognition or models of the world. It just simply requires simple types of combinations of cues. So, for example, let me give you the example of hideability, detecting the opportunity to hide. If you're this lizard and you want to hide because you heard some threatening sound, there are certain patterns of optic information that reliably indicate that this outcropping, let's say here, is a place where you can hide. One feature is a contrast edge, where texture is brighter above and darker below. Furthermore, as you move and approach that object, the motion above the contrast edge should be faster than the motion below the contrast edge. And if that contrast edge lies above the point of visual expansion, then it's probably something you can hide under. And so you don't really have to build a 3D model of the world per se, but just be sensitive to these kinds of combinations of cues as the reptile dorsal cortex appears to be. So I would suggest that these kind of affordance maps developed in this region called the dorsal pallium. Now, this region is important in the story because this will ultimately become the neocortex. So in our ancestors, the synapsids, they retreated from the diurnal world and moved into a nocturnal niche. They became much smaller. They developed warm-bloodedness that allowed this. And they gave up a lot on vision. They lost trichromatic vision. They regressed some of those tactile circuits, and they relied much more on olfaction, audition, and somatosensation. And they expanded this dorsal pallium, which becomes the neocortex, which in mammals has the particularly unusual developmental pattern that's inside out, creating columns, thalamocortical columns, which are much easier in terms of wiring costs to expand dramatically. And of course, it did expand dramatically. Here are some of the other homologs now with more familiar structure: the insula, piriform cortex, and the hippocampus, evolving out of the medial pallium. So, if we look at the brain of mammals, it expanded particularly this dorsal pallial sector, which is organized in kind of a concentric ring architecture, surrounded by lateral parietal regions, which include the orbitofrontal, agranular orbitofrontal, and insular regions, perirhinal cortex, as well as this ring here of, again, agranular cortex, anterior cingulate, retrosplenial, etc. And this central island of 6-layer neocortex, which is subdivided into a somatosensory region, an auditory region, and a visual region. So in sort of sensory specializations, receiving separate inputs from the thalamus, visual input, But also one major innovation of mammals was, of course, that they actually increased the projections from the dorsopallium downstream to take over some of the control of movement. So this somatosensory region became somewhat somatomotor to actually be able to control different types of activity. And another major innovation was a closed loop architecture with the basal ganglia, where the a given part of the somatomotor region projected to the basal ganglia and back again. Now, it's been suggested that this system allows selection of different aspects of the somatosensory region, possibly responsible for different aspects of the animal's behavioral repertoire. So one idea is that the early dorsopallial cortex was organized in these maps for ethologically relevant actions, in particular related to foraging. So searching for food, ingesting food, and all the things in between, which are called handling, which is things like burrowing, breaking open a nut, biting down on something before ingesting it. So specialized systems for these different activities, as well as different kinds of defensive activity, again, driven by visual information. Now, and these could be selected selectively by the basal ganglia, so invigorating different aspects of a behavioral repertoire, depending on your state, your current behavioral state. If you have food in front of you, you work on handling and ingesting. If you don't, you search around. But also visual information was used as an detectum for detecting key stimuli, different combinations of things which may indicate what kind of activity you might want to engage in. And combining that with interoceptive cues from the insula, et cetera, olfactory cues, to develop some sense of the value of different objects that you may be oriented to or sniffing around, and then selecting from these different actions. Now, this is elaborated further in placental mammals with the motor cortex, and then in primates, which returned to diurnal life and vision, rediscovered a color vision, developed large eyes facing forward, relied much more on the visual system, expanding dramatically the temporal, frontal, and parietal cortex, such that the whole thing curls around the insula, creating what we can recognize now as the primate brain, but still organized functionally similarly. So now you have more action maps, especially for these different handling behaviors, all the different things we can do to interact with our world, reach things, bring them to mouth, grasp them, etc., as well as elaborating the key stimulus detection towards full-fledged object classification and recognition, but still in the service of calculating values, etc., for selection. Now, this basic story that I moved rather quickly now can be used to interpret some of that data that I said was confusing. So, for example, why two visual systems? Well, one is used to get information about affordances in the world, the kind of things you can do, specify potential actions, whereas the other one is used to indicate what are the various cues that motivate what you should do right now, how to select those actions through some kind of this sort of ventral stream system. Why should there be multiple representations of space? Well, because each type of activity requires a different representation of space that's reachable, space that's object-related space that's far away, etc. And that we should have these kinds of maps, which has been documented in many animals. Why should variables be mixed? Well, because if there's a continuous competition between different kinds of potential actions that's colored by value, etc., then all those variables should be evident throughout these regions. Why should processing be in parallel? Because you're constantly doing this. You constantly have actions available to you, and you're constantly selecting among them, etc. One example I would want to emphasize is that the gaze control system, because unlike earlier mammals that foraged around by sniffing around and bumping into things, primates could look around, use their sophisticated object system, recognition system, etc., to make decisions about what's useful to approach. The gaze orientation system, because so many of these other activities depend on the high resolution information from fovea, the gaze system takes on a kind of executive role for foraging and organizing your behavior essentially starts to act like what we often call attention, a certain type of attention, which is also, of course, correlated with decision making. Now, what about cognition? Of course, humans can do things that are very abstract. We can play chess and do a lot of thinking before we do any action. So one possibility is that this is a different system, that a cognitive system that developed independently. But I would suggest that it isn't. It actually grows out of basic interactive behavior. So imagine you're this monkey sitting on this branch. You have a berry over here and some potentially appetizing food over here. In your brain, according to this view, these goods are represented in orbitofrontal cortex, about things you might want to approach and ingest and obtain. And perhaps the apples are more appetizing than the berry. But at the same time, your dorsal stream is representing the potential actions. The berry is reachable. This is something you can reach. The branch is something that's walkable. But you can predict, let's say you can predict, maybe through a cerebellar predictive loop, that the berry, the reaching for the berry yields you this good. And that's advantageous. And so perhaps you should not bother walking around. You should just grab the berry. And that's fine. And you can survive this way. But if you're just slightly more sophisticated and have a slightly larger frontal cortex, you can make predictions about predicted affordances. So you know that walking forward makes objects in front, brings them within reach. So if you know if you walk out on this branch, which is possible, then you make the apple reachable. And then another layer of prediction can connect that to the more attractive good. And so now maybe the competition will actually flip. And now you'll decide to forgo the berry and take sort of one step look ahead to go for the apple. Now, it's of course still far from human cognition. You might also ask about language. Well, language, how is language related to this kind of simple interactive behavior? Well, we can actually think of language in terms of, in the same framework that I've been using throughout of feedback control systems, right? So there's control within the body, what we call physiology. There's control through the environment, what we call behavior. But of course, you can also exert control over your state through other creatures. So imagine, for example, you're this helpless human baby and you need some nutrient, but you have no way of obtaining it. It just so happens that in the niche of human babies, there's this thing called a parent. And the parent, with just a simple cue, will do whatever it takes to provide that nutrient for you. And importantly, very complex effects can be obtained with very simple cues that are more or less symbolic. You don't have to tell the parent exactly how to do this, just what it is that you need. And in fact, the parent's going to figure out what it is you need from a very simple utterance. And in time, in fact, actually teach you, essentially through reinforcement learning, what cues to provide to indicate, to try to get these complex effects. So essentially, communication from this perspective doesn't really have to be thought of as a transmission of information per se, that it really exists in a larger context of persuasion. Communication is really persuasion, like this talk. I'm trying to persuade you essentially that this is a useful way of looking at the brain. I think the functional architecture of the brain is the product of a long but discoverable history of adaptations. And I think we can make better sense of it if we understand that history. So really coming back to innateness, there is an innate architecture that we share with animals. And somehow we've learned to do amazing things with it that are quite different than what animals can do. But I think to understand how we do those new things, we really have to know what that architecture was. And I don't think it was, this kind of serial processing architecture that we have in sort of AI and cognitive psychology, but rather a control systems architecture. So it's not serial perception, cognition, and action systems, but parallel circuits for specifying actions, selecting among them, etc. And it really gives us a different conceptual taxonomy. It's not this kind of thing, but rather this, which is kind of like a summary of the talk that I gave, which suggests how different abilities sort of specialized along evolutionary time. And the big advantage of this taxonomy is that this does actually map quite well to the brain, and even very particular subregions of the cerebral cortex, for example. Now, I know I did this rather quickly, so it's probably kind of a blur, but if you're interested, there's some papers published a couple of years ago, as well as a new one that that's in press that probably will be on bioRxiv sometime soon. So with that, I'll stop because I've already gone over my time. Thanks.
Thank you very much for the talk, and we have time for questions. Actually, I wanted to start with a question regarding actually the changes in the brain architecture, because we have this, let's say, MacLean's theory about the trillion brain, that we have this like reptilian brain, blah, blah, blah. So that the next layers were added in the evolution, and it's actually assumed in this model that the older layers are actually not really changed, they're well conserved. We know that this model is wrong, and we actually know nowadays that actually also ancestral circuits can change, and even homologous behaviors in closely related species can have slightly or not even slightly different neural underpinning, because the underlying architecture may change. And in your search in literature, in evolutionary literature, actually, how much support do you find for those two views, like this stability and conservation of, let's say, older circuits and their malleability, the ability of them to change, to be modified in the evolution, even though they are like ancestral ones and related?
Well, there's, to my mind, there is a tendency in the field to revise the sort of the age of structures, usually concluding that they're older than we thought. So if you look at the literature from the 80s and 60s and 70s, you'd look like the neocortex was new, hence neo. Nowadays, no. Nowadays, there are pretty good homologs in lamprey suggesting that some of that basic architecture was already there 500 million years ago. And the tendency often is to discover things are older than we thought. So the conservation is really quite extensive. On the other hand, when you go down different branches, you see very different solutions. So the things that birds do, birds invented, sort of discovered warm-bloodedness, basal ganglia loops, bipedality, tool use, all those amazing things, some of the things that Alison Gopnik mentioned. They developed that independently. There's pretty good evidence that there was nothing like that in the common ancestor. So if you look at a particular lineage, and if you compare to species that diverged, and then the best ones to look at are the ones that didn't change so much since divergence, so things like lamprey, etc. Then you see a great deal of conservation. But if you compare across lineages that are sufficiently distant, you see that very exciting things happen among all of them, but always continuous. And again, development actually, the fate mapping work really establishes just how conservative things have been. Now, there's of course a matter of degree. When I say conservative, I don't mean that things haven't changed. And again, niche transitions are really quite dramatic. The things that happen upon getting out on land were quite dramatic to the structure of the brain.
Thanks. Thanks a lot for this answer. And here one more question, Q&A from Edward Harvey. Some behaviors, such as herding schooling, seem to be necessary for social behaviors to arise, but herding schooling doesn't seem to appear at a particular point in an evolutionary tree, nor does it necessarily remain present in branches. Is herding schooling just a behavior that co-evolves and unevolves repeatedly? How does this relate to innate social factors such as all of our social instincts?
Yeah, so I think that's a good example actually of something that just evolves many times. So if you have an animal that, let's say, becomes herbivorous, a lineage that leads to, that gives up on, let's say, on predation and becomes herbivorous, very often it'll also discover the advantages of herding, etc. So that's very common. In fact, most of the things that you will identify along a particular lineage like human, you'll find along other lineages as well. There's certain fundamental sort of facts of life that animals will discover repeatedly throughout evolution. Vision was, eyes have been invented many times, but very often using the same raw materials, the same photosensitive precursor. So there is a lot of that. And for social behavior, it's important to consider the actual, there's sort of two useful types of lessons. One is about the facts of life, right? What makes a particular behavioral strategy useful and effective for a particular niche, and that we can get a lot out of comparing examples of convergence, like wolves versus primates that looks like evolved their social behavior quite independently, and yet there's some similarities. And so that tells us sort of the problem space that they both are stumbling into and exploiting. But there's the other, the sort of neural mechanistic aspect of how they do it. What are the mechanisms that they use, where you want to look at the actual homology of it. And so you want to look at actually the changes that happen along each of those lineages and led to the particular mechanisms that they have, because they may not actually be the same, even though often they converge to similar structures simply because of kind of like a latent potential that's being exploited. But social behavior, I think for human social behavior, one would want to focus a lot on primates, because there's a lot of good comparative richness there for different social groups that probably are homologous. Social insects are not going to tell us that much, right?
Thank you very much.